Hello everyone, today I'm going to talk about automatic characterization of drug amino acid interactions by energy decomposition analysis. Proteins are the major constituents of our organisms and are involved in almost all processes carried out by our cells, including the reception of drugs. Therefore, the study of drug-protein interactions is crucial to understand the mode of action of those drugs and develop novel therapeutic agents with enhanced efficacy. Nowadays, the application of experimental techniques often remains the most trustworthy approach, but the experimental characterization of a huge number of drug-protein pairs is very time-consuming and costly. Thus, computational methods have gained popularity. But the complexity of these biological systems requires the use of approximations like force fields, which are empirical equations that describe the interaction between particles relating the potential energy of the system with the internal coordinates. So they present physical meaning but low flexibility. That's why machine learning force fields are used. They have a neural network in which you introduce as input the internal coordinates and obtain the interaction energy of the neural network as output, using this general equation. Therefore, they are more flexible but have a lack of physical meaning. That is the reason why we propose a machine learning force fields based on energy decomposition analysis calculation. We first calculate the interaction energy at quantum level. We apply the EDA approach to decompose the energy into its different contributions, and we introduce these terms into the neural network to reparameterize these coefficients. So, when finally you introduce the internal coordinates, you will obtain not only the interaction energy, but the decomposition into repulsion, electrostatic, and polarization terms. So, the use of this EDA approach introduces the physical meaning that conventional machine learning force fields do not have. The first step to develop this force field is to characterize the interaction between drugs and amino acids. In this particular case, we have set up the system for between 250 drugs approved by Food and Drug Administration and glycine. We develop a Python code to perform all the tasks in an automatic way. We compute the energy decomposition analysis and try to find a relation between the EDA components and the chemical structure of the complexes. So, if we are going to calculate the interaction energy, let's define it. The interaction energy between two fragments A and B is defined as the difference between the energy of the complex and the energies of the isolated fragments. The superscript indicates the basis set used to calculate the energies. These interaction energies of the large set of systems have been calculated using semi-empirical and density functional theory methods to decrease the computational time. And then, those interactions have been decomposed into their different contributions by means of EDA approach developed by Professor Marco Mandado. It expresses the energy of the complex as a function of the 1 electron density and the 2 electron exchange correlation densities that are represented as a sum of the unperturbed terms of each of the monomers plus 2 perturbation terms that arise from the interaction of the two monomers. With a little bit of math, this can be simplified in this other expression, where we have the Pauli repulsion terms, electrostatic, dispersion, and induction. The sum of dispersion and induction is the polarization energy. So now let's see the general workflow of the developed code. The goal of this code is to find a geometry close to the minimum energy to compute the interaction energy. For that, first we set an initial distance between the two molecules, then we change the orientation of the molecules to find a favorable orientation for which we calculate the potential energy curve at low level. Later, we calculate the potential energy curve at high level for 5 geometries around the minimum energy. And finally, we apply the energy decomposition analysis approach to decompose the energy into its different contributions. Let's see now in detail all the steps of this workflow. First, we create all the system by combining lysine amino acid with the 250 drugs. For this specific system, composed by glycine amino acid, molecule 1, and this organic molecule, molecule 2, first we calculate the geometric centers of both molecules and separate them by a specific distance. How is this distance calculated? We calculate the Euclidean distance between each pair of atoms. Later, the largest distances for each molecule are searched and called diameters d1 and d2, and half of these values are the radius r1 and r2. Then, imaginary spheres are created around each molecule with a radius of r1 and r2, plus twice the van der Waals radius of the biggest atom of each molecule, to avoid the overlap between atoms. So, this is the distance. Then, we rotate the molecules until obtaining 15 different orientations at the same distance, from which we select the geometry with the lowest PM6 interaction energy to compute the potential energy curve at PM6 level. But how we compute it? Imagine this is the most favorable orientation. First, we decrease the distance 0.1 angstrom until the interaction energy reaches a relative energy of 20 kilocalories per mole, and then increases 0.1 angstrom until the value is 10% of the relative interaction energy at the minimum. Doing that, we obtain the potential energy curve of the system, from which we select 5 geometries close to the minimum, the one corresponding to the energy minimum, 2 consecutive geometries at shorter distances, and 2 geometries every 2 geometries at larger distances. For these 5 geometries, we apply Mobile Tools Kit developed by Gustavo Cárdenas with the contribution of all the members of Mobile Chem Group to create the input files of the complex and the molecule for the interaction energy calculation at TFT level, from which the EDA program will take the electron densities to carry out the interaction energy decomposition in its different contributions, electrostatic, Pauli, dispersion, and induction, along the intermolecular distance. As can be seen, for this particular complex, electrostatic, polarization, dispersion, and induction contributions go to more negative energies when the molecules get closer. On the other hand, the Pauli repulsion term becomes more positive when the molecules approach each other. Now we are going to analyze in detail the results of the total, Pauli, and attractive interactions. Let's start with the total interaction energy. We can see that the interaction energy distribution is relatively broad. That highlights the wide variety of drugs, with functional groups of different nature. We also observe that there are only 11 systems that present a positive total interaction energy. This indicates that the procedure driven by the developed Python codes provides, in most of the cases, a relative orientation and intermolecular distance that lie on a relevant region of the potential energy surface. In the case of Pauli repulsion, it also presents a large broadness, from 0 to 30 kilocalories per mole, with an average value of 9.14 kilocalories per mole. The last terms to analyze are the attractive energy terms, electrostatic, dispersion, and induction. We determine their percentages with respect to the total attractive energy for each system and calculate the distribution of percentages of each of these three terms. In the case of electrostatic, we can observe an average value of 59, 29 in the case of dispersion, and finally an induction energy with a mean value of 12. We can observe that many systems have high polarization, that is not well described by conventional force fields. So, in order to see if there is any relation between the chemical structure of the drugs and the EDA contributions, We analyze the presence of 8 of the most common functional groups present in bioactive molecules and 4 aromatic rings common in the studied drugs. Here it is shown the percentage of systems that present each of the functional groups. We can see that the most frequent functional groups in our systems are group 7, an alcohol, and 8, a carboxyl, for which 80% of the system present these groups, followed by a secondary amine, group 5, with 56% of the population. Finally, the 4 aromatic rings with less than 10% of the population are the least common chemical groups. Moreover, we calculate the average of each attractive energy contribution for the set of systems that present each of the functional groups. We can observe that the predominant term for all of the groups is the electrostatic contribution. Although the percentages are similar along all the set of molecules, the group with the highest electrostatic percentage is group 11, a small ring with a high electron density due to the presence of a nitrogen and a sulfur atom, which can likely participate in strong electrostatic interactions. In the case of dispersion and induction, the drug with the largest contribution are the ones that possess group 6, the one with the chlorine atom, since this atom is easily polarizable due to its size. So, we have seen that the presence of heteroatoms affects the attractive energy terms. Then, the average of each attractive contribution is computed for the set of systems that present a specific number of heteroatoms without taking into account chlorine atoms. It is observed that when the number of heteroatoms increases, the electrostatic term increases, contrary to the dispersion contribution, which decreases, and the induction term is kept almost constant. This can be explained as the larger the number of electronegative atoms, the higher the long-range interactions. In this other case, only considering chlorine atoms, it is observed that when the number of chlorine atoms increases, the electrostatic term decreases, and the dispersion and induction contribution increase, the opposite than in the previous case. Therefore, the nature and the number of heteroatoms present in the drug determine the importance of the different energetic contributions that control the interaction energy between the drug and the amino acid. To summarize, proteins are essential components in our organisms, and the study of drug-protein interaction is very important. Neural networks can describe this interaction, but they need a huge number of calculations. That's why, in this study, we have developed a Python code to automatically create 250 systems, compute their interaction energy, and decompose it in its different contributions using EDA approach. From the results, we have seen that the broadness of this energy distribution indicates that the set of drugs investigated is very diverse. Also, we have observed that the electrostatic term dominates the attractive energy. And finally, we have realized that the heteroatoms have an important role to find an EDA-structure relation. Thank you very much for listening, and see you in the next video.